Good evening, students. Good evening, everyone. Good evening. I welcome you all. I welcome you back. Okay. Today we are going to see the continuation of yesterday's session. Okay. Before going to today's section, I will just introduce about myself. As you all know, myself, Dr. Tamil Selvan, yes. I have completed my undergraduate from TNA Coimbatore and Masters and Doctorate from IRA New Delhi. Okay. I have also cleared CSIR JRF examination, UGC NET, CSIR NET and ASRB NET 2021, 2023 and 2025. Okay. This is just a brief introduction about myself. Now today we are going to see the continuation of yesterday's class. Okay. In yesterday's class, we have seen about only 1 to 60 question we have solved. Okay. Most important previous year question papers we have solved. Okay. Today we are going to see the remaining question from yesterday's question paper. Okay. Students, kindly interact with me. Okay. If you have any doubt, students, kindly interact with me. If you have any doubt, you can ask me at in between itself. Okay. I am expecting an interaction session. Okay. It should be an interacting section. Okay. First question. Okay. Listen the question properly. Read the question properly. First, glycolysis occurs in cytosol. First statement is glycolysis occurs in cytosol. Second statement is citric acid cycle occurs in mitochondrial matrix. Then third statement is oxidative phosphorylation occurs at chloroplast inner membrane. They have given here chloroplast inner membrane. What? Oxidative phosphorylation. They are asking the question, choose the correct answer from the options below. What's the answer, students? Yeah. Svetaraj 1 and 2. Okay. Any other answers? Any other answer, students? Any other answer? Any other guess? What will be the correct answer for this question? 1 and 2. Yes, Tejaswini, it's correct. 1 and 2 is the correct answer. Why? As we know, glycolysis will always occur in cytosol. This is correct option. Citric acid cycle will always occur in mitochondrial matrix. This is also correct. See this option properly. They have given oxidative phosphorylation. Okay, oxidative phosphorylation. Where it will occurs? Oxidative phosphorylation will occurs in mitochondrial membrane. I am right. It will occurs in mitochondrial membrane, not in chloroplast membrane. So this statement is wrong. So the correct answer is option A, 1 and 2 only. Okay. Second question. Which molecule act as an osmoprotectant? Osmoprotectant commonly accumulated under salinity stress. Any guess, students? Which is the correct answer for this? Which is an osmoprotectant? Yeah. Which will be the correct answer for this? Option A, sucrose. Option B, proline. Option C, chlorophyll. Option D, ethylene. Which is the correct answer, students? Yeah, proline. Proline is an osmoprotectant. Proline is an osmoprotectant. The other name of osmoprotectant we can also say as compactable osmolytes. Okay, compactable osmolytes. This is also very important. We can say that as compactable osmolytes. Okay, what this proline will do during the stress condition means this will act as a scavenger. Okay, this will act as a scavenger of reactive oxygen species. Okay, ROS will be produced. That will be scavenged by this proline. Okay, this is also another important role of proline. Okay. This is very important question, students. They have already asked in one time. Okay, which sugar transporter type is involved in apoplastic phloem loading? They are asking here apoplastic phloem loading. What will be the answer? Yeah, Varun Varuni. Yeah, this question they can ask in general agri questions. Okay, general agri portion is there now 1 to 20. In that they may ask. Okay. Students, answer this. Which sugar transporter is involved in apoplastic phloem loading? Any guess? Which one? See the figure also. See the figure properly. Which is the correct answer for this question, students? They are asking about apoplastic phloem loading. Any guess? Any guess? Uday Lakshmi, Tejaswini, any guess? Yeah. H plus sucrose importer. Okay, in the figure itself, it's there. Okay, see here properly. For apoplastic transport, here you see, here there is an H plus sucrose transporter. Okay, this is very important question. Yeah, B is the correct option. Yes, correct. B is the correct option. Next question. Match the questions. Match the students. Match this one and tell me the answer. Read the question properly. First is Rubisco activase. What it will do? Rubisco activase. What it will do? It will helps in maintaining the Rubisco activity by facilitating carbamylation. I am right. This will come for C. What this chlorophyll A and B binding proteins will do? It will harvest light and transfer to reaction center. So, A will have an option 3, B will have option 4. Where it is coming? 3, 4. Here it's coming 3 and 4. Correct? This is the correct answer. Okay. C will have 2. See, plastocyanin. Yesterday class itself we have discussed about this. Yeah, Tejaswini, correct answer. Yesterday also we have discussed about this plastocyanin. Okay. What plastocyanin will do? It will shuttle the electron between cytochrome B6F and plastocyanin. We have already seen. This is PS2. From when light falls on this, electron will be excited. From here, plastoquinone will take. From there, cytochrome B6F will come. From there, plastocyanin will come. This will transfer the electron to PS2. This is clear, students. This is very important. Okay. So, plastocyanin shuttles the electron from cytochrome B6F to PS2. Okay. This is very important question. This they can ask. Okay. Before going to next question, I will just give a brief introduction about nitrate metabolism. Okay. You see this diagram properly. Okay. From here, you will get 1 or 2 questions from this. Okay. Don't miss the concepts. Okay. Listen properly. Don't miss the concepts. Okay. See here. Once the nitrate fertilizer will be applied in the soil. Okay. That will be uptaken by the roots. Okay. Here roots it will uptake. Once it is uptaken by the root, by the action of nitrate reductase, this is important. By the action of nitrate reductase, it will be converted into nitrate in root cells. Okay. The electron source will be NADH. Okay, this is a reducing reaction. So electron source will be NADH. This is also important. Okay. Then this nitrate will enter into the leucoplast. See here, in roots, leucoplast will be there. In leaf tissues, chloroplast will be there. This is important. Okay. In root cell, what it is doing? This nitrate is converting into ammonia by the action of nitrite reductase. The source of electron is what? NADPH from pentose phosphate pathway. This is one of the repeated objective question. They have repeatedly asked this question. Okay. This is the scenario in root cells. This is very important. This is the scenario in root cells. But in shoot, in leaves, what it will happen? Same thing only. Nitrate will be converted into nitrate by the action of nitrate reductase. Here also NADH is the electron source. But you have to observe here clearly. Here for converting nitrate to ammonia, the source of electron is ferredoxine, reduced ferredoxine. This is very important question, students. This is very important concepts also. Okay. Now you solve this question. You read the assertion and reason properly and you solve. Nitrate assimilation requires reduction of nitrate to nitrate in cytosol. Whether it is correct? Second thing, nitrite reductase is the enzyme catalyzing nitrate to nitrate and often uses NADH as electron source. Students, solve the question. Solve and let me know the answer for this. What will be the answer, students? Yeah. Yes, students. First statement is, see here. This is correct. Nitrate assimilation requires that will, what it will do? It will involves the conversion of nitrate to nitrate. But what here if they are saying nitrite reductase is a enzyme catalyzing conversion of nitrate to nitrate. This is wrong. Correct na? The enzyme is, what is that enzyme name? That is nitrate reductase. Correct. That is nitrate reductase. Okay. That is nitrate reductase. So statement, this reason is wrong, but assertion is correct. So what will be the answer? Students, what will be the answer? What will be the answer for this question? Udayalakshmi, Tejaswini, what you guys are doing? Whether you are listening the class, these all are repeated question. I need an interaction. Yeah. Yeah. The answer for this is, yeah, C. Excellent. C is the correct answer. Okay. So statement A is correct. Statement that our reason is wrong. This is the correct answer. Okay. This we have already discussed in our class about the role of plant hormones. Okay, major roles. Okay. Statement 1. Cytokinin delays leaf senescence. Whether this is correct or wrong? That is correct. Gibberellins can break dormancy in some species. This is also correct. Ethylene inhibits abscission in all contexts. Whether ethylene will inhibit? No, it will promote abscission and senescence. So what will be the correct answer for this question, students? What will be the correct answer? 1 and 2. Excellent. 1 and 2 is the correct answer. Yeah, this we have already discussed in our lecture series. Yes. Listen the question properly. Next question. Which enzyme is inhibited by high oxygen concentration? Okay. leading to photorespiration in C3 plants. What will be the correct answer for this? What will be the correct answer for this? High concentration of oxygen that is leading to photorespiration in C3 plants. Which enzyme? Any guess, students? The name of the enzyme itself that have the activity. Which one? Option A, pepcarboxylase. Option B, Rubisco. Yeah, Rubisco. Tejaswini, correct answer. Rubisco. Okay, Rubisco is the correct answer because the name itself it has Rubisco. It has both carboxylase activity and oxygenase activity. This is very important. Okay, this is very important. Next, we are going to see about question on stress physiology. Okay, read the assertion. Heat stress can denature photosynthetic enzyme and destabilize membranes. They are giving, yeah, they are giving heat stress can denature photosynthetic enzymes and destabilize membrane. This is correct statement only. We know because of heat, the proteins may be denatured. So, these enzymes will be proteinous in nature. That will also denature at high temperature. So, this is correct statement. Reason what they are giving? Heat shock proteins. This is an important question. HSP. HSP means heat shock proteins. That will help protect and refold denatured protein under heat stress. Okay. That will helps to protect and refold the denatured protein under heat stress. What will be the correct answer, students? Yes. Yes. Oh, B. A and R are correct. This is correct. Both A and R are correct. Yeah, Uday Lakshmi, excellent. A is the right option. Okay. A is the right option. Why means we know during heat stress condition, there will be a lot of production of heat shock proteins. Okay. The major function of this HSP is to prevent the denaturation of the membranes. Okay. Membrane denaturation, everything will be prevented by this HSPs only. So, both the statements are correct. So, assertion and reason are correct. Then R is the correct explanation of A. This is the right option. Okay, students. While solving this assertion and reason question, you have to think properly and you have to correlate this reason and assertion. Then you choose the options. Okay, whether it is correlating or not. Based on that, you choose the option. Okay, students. This is a direct question. Direct question. They are asking, according to Munch pressure flow hypothesis, the driving force for phloem translocation is. They are asking driving force for phloem translocation. What will be the answer, students? What will be the driving force? Driving force for phloem translocation. What will be the force? It will be ultimately a pressure gradient. Pumping by osmotic loading and unloading. This is a static question. They can ask direct questions only. Memory based. Okay. Next, we are going to see about mineral nutrition. Mineral nutrition is one of the important topic. Mineral nutrition. Okay. You see here, they have given statement 1. Iron is essential for chlorophyll biosynthesis. Iron is essential for chlorophyll biosynthesis. Second statement is what? Iron deficiency commonly causes intravenous chlorosis in young leaf. It's showing intravenous chlorosis in young leaf. Okay, intravenous chlorosis in young leaf. Okay. Iron is mobile in phloem and deficiency appears first in older leaf. Okay, solve this question, students. This concept we have already discussed in our last class. Solve this. What will be the right option? What will be the right option? Iron is essential for chlorophyll biosynthesis. This is correct. Statement A is correct. 1 is correct. Okay. Second statement, iron deficiency commonly causes intravenous chlorosis of younger leaf. This also we saw, this is correct. Then they are giving iron is mobile in phloem. So yesterday itself in class we saw, if a mineral nutrient is mobile, in plant, then deficiency symptom will appears in older leaf. Okay, if a element is, if that is mobile in plant, so deficiency will always happens in older leaf. If it is immobile, if an element is immobile or mineral nutrition immobile, deficiency symptom will happen in younger leaves. We already know iron is one of the immobile element in plant, but they have given here mobile. So this is wrong statement. Then what will be the answer? Yeah, 1 and 2. 1 and 2. 1 and 2 is the right answer. Correct. Can come correct answer. 1 and 2 is the correct answer. Read the questions properly, students. Next assertion. Plants under drought stress often accumulate abscisic acid, which triggers stomatal closure. Okay, this is right, we know. Second question, second reason. Increased abscisic acid in xylem sap, signaling route to synthesis more cytokinin. Whether this is correct? Before going to see this concept, in our hormone class, in our hormone lecture, I have teach about the role of auxin and cytokinin ratio in morphogenesis. Anyone can explain how, when the cytokinin and auxin ratio are same, what it will happen? When cytokinin is more, what it will happen? When auxin is more, what it will happen? Anyone can explain? Anyone can explain, student? Just type in comment box what the high concentration of cytokinin will do, what high concentration of auxin will do, what if the both the ratio of cytokinin and auxin is right? Students, explain this. Explain this. This we have seen in our lecture class. The important role of auxin to cytokinin ratio. Yeah, if same concentration means callus. Correct, Tejaswini, correct. Callus will form. Yeah, callus formation in equal concentration. Yes, Udyalakshmi, right. If high auxin means what it will do? High auxin means root will be formed. I am right. High auxin means root will form. High cytokinin means what? Cytokinin means shoot. High auxin means root. Excellent. See, so here shoot will form. So they are giving, think, if a plant is under stress condition, I already told this concept also. So the root growth will be enhanced, but shoot growth can be arrested or restricted. So here they have given increased ABA in xylem signals to synthesis more cytokinin. Whether this is right? No, this is wrong. Okay, because this increased ABA concentration will helps to synthesis more of auxin. That's why more root growth will be there. If more root growth is there means it will go deeper into the soil and take the minerals and water under the drought stress condition. So this reason is wrong. Okay. So the right option for this is assertion is correct, but or is incorrect. Clear, students? I hope this concept is clear for everyone. I hope this concept is clear. I am proceeding for next question. Which environmental factor most directly influences stomatal opening? The stomatal opening will be directly influenced by which environmental factor? Any guess, students? Any guess? Yeah. The answer for this is what? Light intensity. Light intensity will have direct correlation. This is correct option. How and why? Why light intensity has direct correlation? This is very important concept. See, if light is there, what it will happen? Illumination. That means ultimately illumination will be there. Increased photosynthesis will be there. Sugars will be deposited. Okay. Sugars will be deposited. This will act as an what? This will act in changing the osmotic potential. Changing osmotic potential of God cells. Okay. By changing this, stomatal opening and closing can be controlled. Yes, yes, Uday Lakshmi, correct answer. C is the correct option. So, light intensity is the correct answer. Okay, students. Next question I am proceeding. Given below are two statements. One is labeled as assertion and another is labeled as reason. Okay. First question. Turgor pressure drives cell expansion. Yeah, in the previous concept itself we saw Turgor pressure plays a major role for cell expansion. That is true. Reason. Cell wall loosening by expansions. Cell wall loosening by expansions allow turgor driven growth. Yes, this is also correct. This is a correct explanation of this assertion. So, both A and R are correct and R is the correct explanation of A. This is basic concept, students. That's why I am moving little much fast. Okay. Another question from plant hormone. We have already discussed very clearly about the plant hormones. Yeah. Tejaswani, correct answer. Kenkam, A is the right option. That is correct. Yeah. Yes. Next question. I think some lag is there in this video. That's why answers are coming late for me. What will be the answer for these students? Cytokinin promotes chloroplast development. This we have saw during our lecture series itself. Cytokinin will be involved in chloroplast development. In our lecture, it's there. Auxin promotes apical dominance. Auxin promotes apical dominance. This is also true. We have seen in our lecture. Yeah. 1 and 2. Yes. Ethylene breaks dormancy. No. Then which hormones break dormancy? Students, comment which hormone breaks dormancy? Which hormone breaks dormancy? Yes. Which hormone breaks dormancy? Option A, auxin. Yeah, gibberellin can come. Correct answer. So here, gibberellin should come. Correct. So option 1 and 2 is the right answer. This is another important question, students. This is another important question. Respiratory quotient of aerobic glucose respiration is approximately how much, like that they are asking. They have given option 0.7, 1, 1.3 and 0.5. What will be the answer, students? What will be the RQ, respiratory quotient for aerobic glucose respiration? What will be the ratio, students? It will 1 or 0.7 or 1.3 or 0.5. Yeah, I need your instruction. Yes. Yeah, option B 1. Excellent. RQ will be 1. How it's 1? How it's 1? If you see C6H12O6, if it's react with oxygen molecule, that will lead to the formation of 6 molecule of carbon dioxide. I am right. Carbon dioxide will be formed plus water molecules will be formed. If you see the ratio of oxygen and CO2 consumed, okay, if you see the ratio of oxygen and CO2 consumed, just balance this equation, students, okay. If you see the ratio of oxygen consumed to CO2 released, it will be 1. Okay, that's why we are saying RQ is 1. Then 0.7 is will be for what? 1.3 for what? See, you have to remember one thing. For fat, RQ will be 0.7, and for proteins, RQ will be 0.8 to 0.9. And for carbohydrates, that value will be unity, 1. Okay. And for organic acids, like malic acid, the value will be 1.3. Why I am writing this means, this year 2025 ARS prelims, they asked this question in maths. Okay, that's why I have taken this question. This year they have asked this question. Okay, this is very important. For fat, RQ will be 0.7, and for proteins it will be 0.8 to 0.9, and for carbohydrate it will be unity, that means 1, and for organic acid it will be more than 1 or 1.3. This is clear, students. This is very important question. Next thing, maths. This is very simple question only. You will solve this one. They are giving just an hormone along with that major function. Auxin, it will be primarily involved in apical dominance. Option A should come 4. Abscisic acid, it is a C dormancy hormone. We have already seen. This is a dormancy hormone. So second will should come 1. So this is the right answer. Okay. Then cytokinin, that we know for cell division it will be there. And gibberellins will be involved in stem elongation. This is right. Okay, students. This is about Rubisco only. They are asking again the same question. This will be like similar model only. So I am just skipping this question. Okay. And students, you all know about our coaching institute, AgriAddict Academy. Okay, this is one of the best coaching in India for ICAR PG and PhD entrance examination. Okay. The best thing about our coaching institute is there will be a live as well as recorded online classes will be there. Then we also provides a concise, complete study material. Okay. Nowhere you will get. We will get a concise and complete study material we will provide to you. The most important thing is there will be more than 150 plus mock test series will be there. This will always help for solving the questions during the examination, and there will be a doubt clearing section also will be there. And most important thing, there will be a one-to-one mentorship from all India toppers from IRA. Okay, so kindly register now and get and grab your opportunity, listen the classes and clear the exam. Okay, next question we are going to see. Molybdenum is a cofactor of which enzyme? Molybdenum is a cofactor of which enzyme? Molybdenum is a cofactor for which enzyme? Option A, nitrate reductase. Option B, nitrate reductase, glutamine synthetase and urease. What will be the correct answer, students? What will be the correct answer? Yeah, what will be the correct answer? Molybdenum is the cofactor for? Yes, Uday Lakshmi. Tejaswini. Yeah, 4321 is right option for last question. Tejaswini, correct. Molybdenum is cofactor of which enzyme? Yes. Option A. Nitrate reductase is the that will have molybdenum as a cofactor. If you see the structure of this nitrate reductase, okay. If you see the structure of nitrate reductase, there will be a 3 component. Okay, 3 component will be there. Okay, this is nothing but electron carriers. Okay, first FAD domain will be there. Okay, FAD domain will be there. Then what will be there? molybdenum domains will be there. Okay, molybdenum iron sulfur clusters also will be there. Okay, so we can see in the structure itself it's there molybdenum. So only nitrate reductase will have molybdenum in its structure. It's clear, students. Next question about photosynthesis only. We will solve more photosynthesis question. Okay, this is one of the important topic. That's why so much of questions are there from photosynthesis. Okay, C4 plants. Statement 1 is C4 plants have higher water use efficiency than C3 plants. Yes, this is correct. C4 plants lacks Rubisco. C4 pathway involves Kranz anatomy. This is correct. They are asking here, they are telling here, C4 plants lacks Rubisco. But if you see the C4 cycle, there will be a Kranz anatomy in plants. Okay, C4 plants. Kranz anatomy means what? There will be a here mesophyll cells will be there. Bundle sheath cells will be there. Okay. This is just a mechanism of concentrating the CO2 in particular organelle or particular tissue to enhance the efficiency of Rubisco. Okay, you see here, atmospheric CO2 is coming. This will be converted into what? Bicarbonate. This bicarbonate by the action of PEP carboxylase, that will be converted into oxaloacetate. This will be converted into malate. This will go here. Okay. So see here, first carboxylation is happening by PEP carboxylase. Okay. But after entering to the bundle sheath cell, it will release the CO2. The malate will be converted into pyruvate and CO2. Okay. Phosphoenol pyruvate and CO2. Okay. Phosphoenol pyruvate and CO2. Okay. This will be fixed by what? That released CO2 will be fixed by which enzyme? Rubisco only it will do there. So Rubisco will be present in where? Bundle sheath cells of C4 plants. But in questions they are saying Rubisco is lacking in C4 plants. So this is wrong answer. This is wrong. Yeah, 1 and 3 is the correct. Excellent, students. 1 and 3 is the correct. Solve this question, students. What happened? Because of that one only. Unmuted. Answer for this, students. Answer option A. Both A are correct and R is correct explanation of A. Yes, we have already discussed about this. Okay, we have already discussed about this. Both A and R are correct and R is the correct explanation of A. Thank you, students. Next question. Which complex containing magnesium cluster? See here, manganese cluster which will have responsible for water oxidation. Understand the question? Manganese clusters will be there. That will be responsible for oxidation of water. Where oxidation of water will happen? Where? This is one of the repeated question. Students, solve this. Yeah, B. PS2. Correct answer, student. In PS2, okay, exactly if you want to see here, photolysis of water will happen. Why? Why photolysis of water is happening? To fill the electron gap in PS2. Okay, to fill the electron gap in the PS2. So that will have a manganese cluster. So this will be the right option. So photosystem 2 will be the right option. Next question. This is just a simple question. We already know glycolysis end product is what? That will be pyruvate. Glycolysis end product is pyruvate. So option A will come 1. TCA cycle. It is also known as TCA cycle is also known as what? TCA cycle is also known as citric acid cycle. Okay. The end product of that one is what? End product. That will be what? End product? Carbon dioxide and NADH. End product will be there as carbon dioxide and NADH. So B2 fermentation. Fermentation means anaerobic respiration. There the end product will form ethanol and CO2. Yes. C3 and D. Beta oxidation means what? It's a sequential removal of 2 carbon compounds. Tejaswini, correct answer. 1, 2, 3, 4. Yeah. Tejaswini is correct answer. Okay. So beta oxidation means acetyl CoA will be released. That will be the end product. Yes, students. This already we have seen in our yesterday's lecture about the phytochromes. Okay. Phytochromes mediate response show red and fair red reversibility. Yes, this is right. We already saw in yesterday's lecture. Fair red light converts PFR to PFR. PR inactivating the response. What will be the correct answer? What will be the correct answer? They are saying PFR is converting to PR when fair red light is there. This is true only if fair red light is there, PFR will be converted into PR form. This up to this is correct, but they are saying PFR is inactivating the response. That's correct. PR is the inactive form of phytochrome. Correct. So both A and R are correct. Yeah. Both A and R correct. And R and Uday Lakshmi. Option A is right answer. Okay, not option B. Okay, both A and R are correct and R is the correct explanation of A. Okay, any doubt in this? Any doubt? Because fair red light convert PFR to PR, inactivating the response. Any doubt, students? Yeah, I am proceeding. Which antioxidant enzyme is localized mainly in the peroxisome? They are asking which antioxidant enzyme mainly localized in peroxisome. This is very important question. Okay, you have to know the marker enzyme for each and every organelle. Okay, each and every organelles will have a marker enzyme. Okay, so marker enzyme means what? That enzymatic activity will be particularly present in that only one organelle. For example, if you see catalase, okay, catalase will be present only in peroxisome. Okay, only in peroxisome. Okay, another example, anyone can tell what is the marker enzyme for endoplasmic reticulum? Can anyone tell what is the marker enzyme for endoplasmic reticulum? Marker enzyme for mitochondria? Marker enzyme for glyoxysome? A marker enzyme for these things is enough. Can anyone tell the examples or marker enzyme for these organelles? Students, this is your homework. Okay, you just after this lecture, you just go and see. This is one of the important question. They can ask what are the marker enzymes? What are the marker enzymes for individual organelles? Okay, you are going to appear for ICAR JRF examination. They will test your basic knowledge about organelles also. Okay, the right option for this is catalase. Okay, you just go and see about this. Each and every organelle marker enzyme is important. Okay. Next question. Transpiration helps to cool leaves. Okay, read the question properly and let me know if any doubt is there. Transpiration helps cool leaf. This is true. Transpiration aids uptake of minerals. This is also true. Transpiration always reduces photosynthesis. This is wrong because we know transpiration is a necessary event that will be happen in the leaf. Okay, it will not reduce the photosynthetic efficiency. Okay, so that is a necessary event. So this third statement is wrong. So option 1 and 2 is the right. So statement 1 and 2 is the right answer. Yes, Tejaswini, this is correct answer. 1 and 2 is the right. Okay. See, next question. Nitrification is the biological conversion of ammonium to what? What? Nitrification? Nitrification is biological conversion of ammonium to what? What? Nitrification. Option A, amide. Option B, nitrate. Option C, nitrogen. Option D, urea. Nitrate. Option B, nitrate. This is right answer. Before going to next question, I will just give a brief introduction about the definitions in nitrogen cycle. Okay, what is mean by nitrification? See here, listen properly. Okay, we are applying nitrate to the soil, nitrate fertilizer we are applying. That will be converted into nitrate. This nitrate will be converted into nitrogen gas. This process is known as what? Denitrification. Okay. Denitrification means what? Conversion of nitrate to nitrogen gas. This is known as denitrification. Okay. This conversion of nitrogen to ammonia. Okay, nitrogen to ammonia. This we can say ammonification. Okay, ammonification. Okay. Then denitrification means what? Conversion of this ammonia back to nitrate. This is known as nitrification. These three concepts are very important, students. Denitrification is the conversion of, question what they are asking. Denitrification is the conversion of ammonium to what? Conversion of ammonium to nitrate. So this will be the right answer. Okay. This is the right answer. This you have to learn this. Okay. Just remember, they may ask about these definitions also. What is denitrification? What is ammonification? And what is nitrification? They may ask these questions also. Okay. Next question. Given below are two statements. Okay, one is assertion and reason. Just solve this question. What they are asking? Abscisic acid levels rises in leaves under water deficient condition. They are saying abscisic acid level rises in leaves under water deficient condition. This is true. ABA is a stress hormone. Under stress condition, this synthesis may be enhanced in leaves. Second option. Abscisic acid transport from root to shoot signals closure of stomata. This is also correct. So, what will be the correct answer, students? Excellent. Both A and R are correct, and R is the correct explanation of A. Yes, correct. Can come correct answer. Yes. Next, this is very simple question, students. You will solve. I know. Okay. Magnesium will be an important component for. Magnesium is an important component in chlorophyll. So A will come 1. Where A is coming 1 here, so 3 is okay. Iron is an important component of what? Before going this, we already saw nitrate reductase will have molybdenum as a cofactor. For D, option 3. Where it's coming? D, option 3. Okay, here only coming. So this is the right answer. See how easily we are solving the questions. If you know one or two options means we can solve. But for clarity, yeah, D is the correct answer. Yes, correct, correct. For clarity, we have to recheck once. Okay. So for A is 1, okay, A1, B2, this is correct. Copper, we already discussed in yesterday's class. Copper is a cofactor or enzyme component in plastocyanin. Excellent. Yesterday we have saw this concept. So plastocyanin means cyanine will have copper. Okay. Very good, students. Next question. This is very important concept regarding electron transport chain. Okay, very important concept. Properly you read the question. ATP synthase is localized in thylakoid membrane. They are saying ATP synthase will be localized in thylakoid membrane. Yes, it's true. This is a chloroplast. Here granules will be there like a stack of coins. Here ATP synthase will be there. Okay. ATP production is driven by proton gradient across thylakoid. Yes, this is also correct. Would Kenkam already solved the question. Option A is right. Okay. We will see this. Third thing, stoma has high H plus concentration than lumen during illumination. No, this is wrong answer. Okay, so option A is right. 1 and 2 is correct. Everyone answered correct answer. 1 and 2 is correct. Yeah. So, which will have high H plus concentration? Lumen will have high amount of H plus concentration. Okay. Just you imagine this one, then concept will be clear. Okay. See, this is a thylakoid. For example, this is the chloroplast. This is a thylakoid membrane like a stack. It will be there. Like this, a stack. Okay. Light reaction will happen here. I am writing here light reaction. Okay. So during light reaction, what will happen means the H plus from the stroma, H plus from stroma will be pumped inside to lumen, thylakoid lumen. Okay. So H plus gradients will be formed. Here in thylakoid, in the thylakoid, ATP synthase complex will be there. ATP synthase complex. What it will do? It will pump the H plus ion back to the stroma. Because of this process, ATP will be synthesized. This will be utilized in CO2 fixation. Concept are clear, students? So during illumination, that means during light, H plus concentration will be more in lumen. Okay, comparing to stroma. Next question. Which is the primary substrate for Rubisco oxygenase reaction? Rubisco oxygenase reaction. Students, answer please. Yeah, which will be the correct answer? Yes. Ribulose 1,5 bisphosphate. Ribulose 1,5 bisphosphate is the right answer for this. Next question. Cytokinin delays leaf senescence. This we already saw in our class. Cytokinin delays leaf senescence. Yeah. A, B, C. Okay. Then they promote mobilization of nutrients towards leaf. Yes, Tejaswani. Yeah. Towards leaf and chloroplast maintenance. So the both A and R are correct, and R is the correct explanation. We know cytokinin will delays leaf senescence. This concept we have already discussed in our previous lecture. This is known as Rickman-Lang effect. This we already saw in our lecture. In our lecture slide itself, it's there. If you go and see that, we know cytokinin will also involved in mobilization of nutrients. This also we have already discussed in our class. So assertion is right, reason is the correct explanation. Which enzyme catalyzes the link between glycolysis and TCA cycle? Which is glycolysis and TCA cycle link? What is the correct answer, students? What is the correct answer? Which enzyme catalyzes the link between glycolysis and TCA cycle? Yes, any guesses? Which one? Pyruvate kinase? Pyruvate dehydrogenase? Can come right option. Pyruvate dehydrogenase. We can see that as PDH complex. Why we are saying this as a complex means in this it will have 3 enzymes. Okay. Okay, 3 enzymes will be there. This is important. Okay, 3 enzymes will be there. That's why we are seeing this as pyruvate dehydrogenase complex. Okay. Okay. So this will be act as a link between glycolysis and TCA cycle. Okay. Read the again photosynthesis question only we are going to see today. Okay. CAM plan show diurnal acid fluctuation. Yesterday itself we saw this is correct because malic acid will be accumulated in the vacuole during night time. Okay, during daytime this malic acid will be utilized. Yes, C4 plants perform spatial. Why I have took this question? I will explain you. Here, spatial separation of CO2 fixation and reduction. Okay, spatial separation is there. Then C3 plants have high photorespiration under light. All the statements are correct. Correct. Why we are going to see about spatial. What is the difference between spatial and temporal separation? We already discussed about this concept. Spatial means, yeah, 1, 2, 3 is correct. Okay. Spatial means separation between two organelles. For example, mesophyll and bundle sheath cells. Mesophyll and bundle sheath cells. That is, we can say as Kranz anatomy. Kranz anatomy will be there in only C4 plants. But in CAM plants, there will be a temporal separation. There will be a temporal separation. Temporal separation will be there. So option is 1, 2, and 3 is correct. Very good, students. Again, important question. What's the difference between SIMPO transporter, antiporter? Just match this one and let me know the answer. Match this one and let me know the answer. Option B. Okay, Udayalakshmi, option B. Okay. Udayalakshmi, option B. Then anyone else? Anyone else? What is the answer? Yes, Preeti Kaur A. 2134. Okay. 2134. Okay, this one. Okay. Yes. Everyone said correct answer. Correct answer. A is the right answer. You see this concept properly. Okay. Symport means what? Symport means that's a movement of two ions or two molecules in the same direction. This is symport. Okay. For example, here one transporter is there that is transporting two ions, ion 1, ion 2 in same direction. Okay. Antipode means what? Antipode means opposite direction. One is going outward, one is coming inward. Okay. Third means what? Uniport. Uniport means what it will do? A single solute in one way. That may either it may go out or either it may come in. Okay. Aquaphorine. Again, I am saying this is important question. This they have asked during this time prelims examination for us. Okay. ARS 2025 they asked. Aquaphorine, what it will do? It will facilitate water movement. It will facilitate water movement. Okay. You have to learn about basics also because from basics sure you will get questions. Okay. So this is the right answer. Students, this is another important question. This also we have already seen in our class. Deficiency of sulfur affects pollen tube growth. They are saying sulfur deficiency affecting the pollen tube growth. Okay. Molybdenum is an important for cell wall forming and membrane integrity. Whether these both statements are correct, what will be the correct answer for this? If it's wrong, what will be the correct answer for this? Just type in comment box, students. Kenkam, Tejaswini, Udaya, any doubt? Just type the answer. Yeah. Yes. Yes. Deficiency of sulfur. Whether sulfur will cause the pollen tube growth will be affected. No. A is incorrect and B is also okay. Udaya Lakshmi is saying A is incorrect and B is correct. Okay. So A is incorrect. This is correct. Okay. Because deficiency of sulfur will not affect pollen tube group. Boron. Yeah, boron can come correct. So boron will be important for pollen tube growth. Pollen tube growth. So this is wrong. Assertion is wrong. Then next question. Molybdenum is important for cell wall formation and membrane integrity. Which one is important? Whether molybdenum is important? Which is the correct answer? Molybdenum is not involved in cell wall formation and membrane integrity. Which one? Yes, option A, calcium. Option B, sulfur. Option C, iron. Which is the right answer, students? Yes. Udaya Lakshmi, which will be the right answer? Magnesium. No. I didn't give option magnesium. Calcium, yes, Kaur. Calcium is correct answer. So calcium is important. Okay, calcium is important for cell wall formation and membrane integrity. This is important, student. Calcium, yes. Both are incorrect. Calcium is, yeah, calcium. So calcium is important for what? Cell wall formation. Why? Why, sir? Why calcium is important? Not magnesium or these things. If you see, the middle lamella of pectins will be there. You know pectin. There will be calcium pectates will be there. That's why calcium is important for cell wall forming. Understand, students? Next thing, which small molecule act as universal second messenger in many abiotic stress? Read the question properly. First grasp the question what they are asking. Then you go and see the options. Okay. Which small molecule act as universal second messenger in abiotic stress? Students, any guess? Any guess? Yes, any guess? Option A, ATP. Option B, calcium. Option C, nitrate. And option D, NADH. Any guess? Yeah, any guess? Yeah, calcium is the right answer. Okay, calcium will act as a secondary messenger during abiotic stress. Yeah, calcium, correct. Calcium is the correct answer. This everyone will solve. What is the first stable product of Calvin cycle? It will be what? It will be what? What will be the stable product of C3 cycle? Students, what will be the first stable product of Calvin cycle? Comment your answers. Yes. 3-phosphoglycerate. Option A is the right answer. 3-phosphoglycerate. Given below a two statement. Yeah, A is the right answer. Yes. Given below a two statement, see. Camp plants open stomata at night. This already we saw. This is right statement. They store carbon dioxide as malic acid to minimum water loss. This both are correct. Both are correct. Huh? Whether this both are correct. Option A is correct. Assertion is correct. Reason, whether they will store malic acid to minimize the water loss. Huh? To minimize the water loss, whether they will do, no, they will open the stomata during night time. Open stomata, they will open stomata during night time. during night time to prevent the water loss. So only one statement is right. A is correct, but R is incorrect. Understand this, students? Next question. Which hormones break seed dormancy by stimulating alpha amylase enzyme activity in barley? Students respond. Which one? Which one is breaking the dormancy by stimulating alpha amylase synthesis? Stimulating alpha amylase synthesis in barley. Answer, students. Type your comment. Which will be the correct answer? This very simple question. We have already seen this one in class. Yeah, gibberellins. Okay, gibberellins is the correct answer. It will promotes alpha amylase activity. So breakdown of starch will be there. That will stimulate the germination of the seeds. Okay. Again, another questions from plant hormone portion. This all these things we have seen in our lecture. If you miss the lecture, just go and rewind. Okay, just go and see once. Okay. Ethylene statement one is ethylene induce epinasty. Okay, ethylene induce epinasty. Okay. Second, ethylene enhance fruit ripening. Statement 2. Third is ethylene delay senescence. What will be the correct answer, student? While taking our lecture, someone asked about what is meant by epinasty also. I still remember. Someone asked what is meant by epinasty, sir. Yeah. 1, 2. Yes, 1 and 2 is correct. 1 and 2 is correct option. It will, ethylene will enhance senescence. Okay, so 1 and 2 will be the right answer. Yes, Tejaswani, correct answer. This everyone will answer. This is just a basic which organelle will be involved in which synthesis. For example, glycolysis will be involved in, will be occurs in cytosol. So one will have C. This is the right answer. Nothing will have other options. So this is the correct answer. A is 3. Glycolysis will happen in cytosol. B is 2. TCA will happen in mitochondria matrix. Calvin cycle will happen in stoma of chloroplast. Photorespiration will happen in where? Peroxisome, chloroplast, and mitochondria. If you see the organelle sequence, chloroplast, peroxisome, mitochondria. CPM, chloroplast, peroxisome, and mitochondria. 3214. Yeah, correct. 32. Udaya Lakshmi, correct. 3214. Yes. D option. Yes. Students, next question we are seeing. Okay. Blue light activates phototropin receptor. We already seen this one in last lecture. Yes, blue light will activate the phototropin receptor. This is true. Phototropins mediate stomatal opening and phototropic curvature. This is also true. In yesterday lecture we saw. So what will be the answer for this? Both A and R are correct, and R is the correct explanation of A. Okay, this we already saw in our yesterday's lecture. That's why I am going very fast. Okay. Again, an important question. Dieback of shoots. Deficiency of which nutrient? Deficiency of, yeah, deficiency of which nutrient causes dieback of shoots in many plants? Dieback of shoots in many plants. Which one? Deficiency of calcium. I'm right. Yeah, correct. Option A is correct. Calcium. Calcium will cause dieback in shoot tips. This is very, very important question. Calcium will cause dieback. Yes, calcium. Yes. Next, translocation in phloem is bidirectional. Which is the correct answer? You just solve and let me know which one is the correct answer among this. Xylem transport mainly water and mineral nutrient unidirectionally. Phloem uses much pressure flow mechanism. Which will be the correct answer for the students? Which will be the correct answer? Yes. You read the question properly and let me know which will be the correct answer for this. D, sir. All are correct. Yes. Excellent. Okay. Why here bidirectional is there? Why here unidirectional is there? As we know, xylems will be primarily involved in translocation or uptake of minerals and nutrients from roots. Then it will distribute to shoots. This will be always unidirectional. But if you see for phloem, it is bidirectional. Why? Why it is bidirectional? As we already saw, during the senescence phase, the nutrients that are present in the leaf or shoot or wherever it may be, that may be remobilized or relocalized to other parts of the plants. So there will be a bidirectional transport. Okay, that's why translocation in phloem is bidirectional. This is clear, students. Read by concepts. Okay, D is the right option. Rubisco is the most abundant enzyme on the earth. Reason, it catalyzes oxygen fixation in Calvin cycle in all autotrophs. Which is the correct answer, students? Yes, Rubisco is most abundant enzyme on earth. It catalyzed oxygen fixation in the Calvin cycle in all autotrophs. Which will be the correct answer? Which will be the correct answer? Yes. C. Okay. C. Okay. Yes. Yes. C is the right answer. Because why? It catalyzes oxygen fixation. It will catalyze CO2 fixation in the Calvin cycle. So, C is the right option. Assertion is correct, but R is not correct. Yes. Which cofactor is required for functioning of nitrogenase enzyme? Nitrogenase enzyme. Don't confuse between nitrate reductase or nitrate reductase. What will the answer for this? C, sir. What will be the answer? B. Option B. Yes. Molybdenum ion complex will be present in nitrogenase. This nitrogenase will be involved in biological nitrogen fixation. As we know, this is very important concept, students. This they asked in 2019. I wrote my MSc in 2019. That time they asked this question. Okay. Number of ATP molecules required for nitrogen fixation, per molecule of nitrogen fixation. How much it will be there? Option A 12, option B 14, option C 16, option D 18. What will be the answer for this? Number of ATP molecules required to fix one molecule of nitrogen through biological nitrogen fixation. Yes, students. 12, 14, 16. Yes, excellent. Kenkam, 16 is the right answer. Okay, this is very important point. 16 ATPs will be required for biological fixation of one molecule of nitrogen. Okay. Udaya Lakshmi, yes, everyone correct answer. Next question. This we already saw. Just simple question solve, students. This concept we already saw. Respiratory quotient is less than 1 for fat respiration. Statement 2, RQ will be greater than 1 for gibberellic acid. They are saying gibberellic acid respiration, RQ will be equal to 1 for carbohydrate respiration. What will be the answer, students? What are the statements correct? What are the statements are correct? Yes, students, yes. Which will be the correct? Oh, which statements? Yeah, all are correct. Udaya Lakshmi, excellent. All statements are correct. Okay. This we already discussed in previous questions. Okay, RQ will be 1 for carbohydrates. Just we are remembering now. RQ will be 1 for carbohydrates, that is unity, 0.7 for fat, 0.8 to 0.9 for proteins, and greater than 1 for organic acids. Okay. Yes, 1, 2, 3, correct. Yes. Next question. Plastocyanin transfer. This already we saw this one. This is simple question about light reaction. We already saw about this question. Okay. Plastocyanin transfer electron between photosystem 2 and cytochrome B6F. Okay, this is correct. It is a lipid soluble carrier in thylakoid membrane. It is a lipid soluble carrier. What is the correct answer? It's a lipid soluble carrier. Yes. Yes, 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 yes. Both A and R are correct, and R is the correct explanation of E. Correct, students. This is the correct answer. Plastoquinone is a lipid soluble carrier that will be present in thylakoid membrane. Okay. Next question. When cell is placed in hypertonic solution, what it will happen to the cell? What it will be happen to the cell? This is important. Okay, you just understand the concept. If a cell is placed in hypertonic solution, what it will happen? It will placed in hypotonic solution means what it will happen. If it's placed in isotonic solution means what it will happen. Okay. If a cell is placed in hypertonic solution, the membrane will be busted due to, yeah, plasmolase. Correct, correct. This we can say as plasmolase. Okay. So the cell will be busted, that will lead to plasmolysis of the cell in hypertonic solution. But if we placed on hypotonic solution means what it will happen? Water will come inside to the cell. So cell will become turgid. Okay, turgid. This will come for hypotonic solution. Okay. Flaccid will come in isotonic solution. Okay. So we can frame 3 questions from these options. Okay. So the right option for hypotonic solution is plasmolized. Okay. This we already saw in our lecture. See, all the questions we have already worked out in our lecture. Auxin. Who has given the name auxin? Auxin. Gibberellin, cytokinin, abscisic acid. All the names we have discussed. Solve this question, students. Solve this question. Yeah, options. I need just options. Which is correct, which is not correct. Yes. Option C. 1, 2, 3, C. Yeah. A option, sir. 1, 2, 3, 4. Yeah. Auxin was given by FW Wendt. FW Wendt. Gibberellin, Kuroshwa. Cytokinin, Miller. Abscisic acid, Adicard. This is right option. Very good. Very good. Very good. Just another simple question. C4 plants, we already know. Grant's anatomy will be there in C4 plants. This is true. This you see again. Bundle seed cell possesses large chloroplast, lacking grana. Lacking grana. Before going to see that, first you have to tell the function of grana. What grana will do? What's the function of grana? What it will do? Always you have to go by concepts. Then only we will never forget. Don't mug up even a single question. Go by concepts. Okay. We already know in grana only light reactions will happen. Okay, light reaction will happen in grana. This we already know. Okay. In stroma, what it will happen? Dark reaction will happen. Dark reaction means what? Nothing but CO2 fixation will be happen. If you remember about, yeah, light reaction, yes, excellent, excellent. Yeah. If you remember, the thylakoids will have granas. Okay, membranes of granas will be there. Okay, yeah, granas will be there. They have given in bundle seed cell, larger chloroplast will be there, lacking grana in C4 plants. This is right answer. Why I am saying means, see, this we already saw in our class. Okay, today only we saw. There will be 2 cells. One is what? Mesophile cells will be there. Another is bundle sheath cell. Okay. See, just imagine. Okay, just imagine one concept. Okay. This is very important. Just imagine one concept. Okay. This is bundle sheath cell. Okay. This is bundle sheath cell. Okay. Around this one, mesophile cells will be there. Okay. Mesophile cells will be there. This is known as Kranz anatomy. Kranz anatomy. Why, sir, this bundle sheath cell has to be protected by mesophile cells? We already know this Rubisco enzyme will have both carboxylase and oxygenase activity to prevent the reduced oxygenase activity and to enhance the concentration of CO2 in this specific bundle sheath cell. Okay, in this specific bundle sheath cell, this anatomy is there. That's why, see, this mesophile cell itself will produce enough amount of ATP and NADPH required for dark reaction. So, this bundle sheath will always possesses large chloroplast lacking grana because light reaction no need of happening in the bundle sheath cell. That's why both A and R are correct and R is the correct explanation of A. Okay, students. ATP synthase enzyme complex is also known as. This is very simple, students. Just answer. Which is the correct answer? ATP synthase enzyme complex is also called. Yeah, option A, correct, correct. Can come correct. CF0 or CF1 complex. Yes, 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 correct one. Next question. This is very simple question. Abscisic acid induced stomatal closure. This we already saw. This is true. Abscisic acid accumulate under saline stress. True. Abscisic acid stimulates seed germination. What will be the correct answer for these students? Whether abscisic acid will stimulate? No, abscisic acid will inhibit. It will inhibit seed germination. That's why we are saying abscisic acid is antagonistic to gibberellic acid function. ABA is antagonistic. Antagonistic to gibberellic acid. So this third statement is wrong. So 1 and 2 is correct. This is clear, student. 1 and 2 is correct. This is another question on photosynthesis. This will be easy only. You will do this one. Okay. Next question. Movement of ions against electrochemical gradient. What will be the answer? Movement of ions against the electrochemical gradient. So against means what? From higher concentration, from that is moving against. For example, here more concentration is there, here less concentration is there. But against the concentration it moving means that is active transport by utilizing ATPs. Okay, ATP will be utilized. So active transport. Next question. Mitochondria, yeah, correct. C is the right answer. Mitochondria and chloroplast both have electron transport chains. Yes, this is correct. ATP synthase both uses electron transport. That is also correct. Yes. Both generate CO2 as byproduct. Whether this is correct? Both generate CO2 as byproduct. No. No. What will be the end product? What will be the end product? What will be the end product? Water will be the end product. So option C, wherever option C is coming, it's wrong. So 1 and 2 is the right option. Yes, Kenkam, C is the right option. Hydrathodes, this is a simple concept only. Everyone knows about what is meant by gutations. Okay, guttation means what? It's an exclusion of water from the leaf tips during early morning period. Okay. So see, that will be done by hydrathodes. Okay. Hydrathodes are, that means exclude xylem sap during gutations. This is true. This is basic concept. Everyone knows. Second, root pressure forces liquid water and out the. This is also correct. This is a simple concept. Yeah, correct. Option A is correct. Kenkam, option A is correct. Ammonification. Yes, I have given this. Yeah, ammonification. What? Yes, correct. Tejaswini Udalakshmi, correct. A is correct. Okay. Ammonification is conversion of dash. Ammonification. Option A, nitrate to nitrite. Option B, organic nitrogen to ammonium. Option C, ammonium to nitrogen. And option B, nitrate to nitrate. What will be the correct answer? Yes. B, organic nitrogen to ammonium. Excellent. Then what will be the conversion of nitrate to nitrite? What will be the conversion of ammonium to nitrogen? Can anyone tell? What is the, okay, this we already saw the definitions. Just to go through once. Okay, this is important concept. Okay, definition they may ask. Phytochrome statement, three statements they have given. Phytochromes are chromoprotein. This we already saw in our lecture. Yeah, it's a chromoprotein. Correct. It absorbs both red and infrared light. Yes, this is also correct. PR forms absorb fair red light maximally. Which is the correct answer? About notification, we will let you know. Okay, we will let you know once it comes. Huh? So the PAR form observes red light, not fair red light. I am right. So option 1 and 2 is correct. Okay. Okay, students. Option 1 and 2 is, yeah. Tejaswini, 1 and 2 is correct. Last question of today's session. And that too one of the most important question from nitrogen metabolism. Students, solve this. Read the question properly. Biological nitrogen fixation is a energy intensive process. Reason, each molecule of nitrogen reduced to ammonia requires at least 16 ATP. What will be the correct answer for this, students? What will be the correct answer for this? Yes. Yes. Assertion is biological nitrogen fixation is a energy intensive process. Yes. A is the right answer. We already discussed this. Biological nitrogen fixation is an energy intensive process. It requires 16 molecules of ATP to reduce nitrogen to ammonia. Yes, everyone correct answer. Correct answer. Correct answer. Yes. A is the correct answer. So both A and R are correct and R is the correct explanation of A. That's all, students. Today's session is over. Okay. Thank you all for your patient listens. Okay. Tomorrow we will see in another session. Okay. Thank you, students. You may leave now. Guru.